Why did you say that?
All right, welcome back from fall break. I hope you had a good break. Let's see. I read in that last question on the exam that a lot of you were seeing family or traveling or just hanging out with friends. So sounds like a lot of people had some exciting breaks. I did not go anywhere. I just got some household chores done, but for me that was pretty exciting. To have time to do that. So I passed back exams. You'll notice that some of them I put like an okay and I didn't take any points off. One of them being that table where I asked you like the specific numbers of energy-containing molecules. A lot of you forgot to double, but you got the right like numbers for a single molecule of glucose. So I knew that you knew it. You just forgot to double. So I did not take any points off of that. There were a couple, let me change the slide here. Class average was actually really great. Last year, the class average was a 77%. So I don't know if it was that shorter timeline that made you like try to study a little bit more than my last year where they had more class periods. Not sure, but it was fantastic. I was impressed by that. There are some that you can get some points back for, right? There were two that, if you got that question wrong, I automatically gave you points back because I felt like maybe the question I asked was oddly phrased, it was unfair, there was something happening there. Let's see. So one of those questions is 3B, and that is the one talking about the enzyme. I really wanted the answer, like for you to come to the answer that it's an enzyme, so it's a catalyst, it speeds up a reaction, makes the reaction go faster, something along that lines. A lot of you wrote that it converts fructose 6-phosphate into fructose 1,6-biphosphate, which yes is correct, but isn't the purpose of like enzymes in general. I just felt like I worded that wrong. I gave you the point back if you got that wrong. The other one is short answer 6D. This was about the that protein that allowed protons to freely diffuse across the membrane, right? And how would that affect your oxygen usage? I was hoping that you would get the answer that it stays the same, right? That protein has absolutely no effect on how oxygen is used. Right? Electrons are still being donated, electrons are still going through the electron transport chain, oxygen still has to collect the electrons. Hydrogens, they're now freely flowing across the membrane, right? So they're still around, they're still able to connect with the oxygen to make water, right? They're just not building up in concentration to go through ATP synthase. Some of you gave me a different answer, but a very good reasoning behind that answer. I gave you the point. And then if I marked it wrong, I gave you that point back as well. Right? Because again, I just felt like those were weird questions and that so many of you got them wrong that I think there was something on my end that maybe I didn't elaborate enough on. There are some opportunities for points back. So that's going to happen same manner as before. So don't erase the incorrect answer, correct it on another piece of paper, write me the rationale. I linked the full instructions that are on Moodle. They're on Moodle under Unit 2 since it's a Unit 2 exam. So if you're looking for them, that's where they're hidden. I won't say they're hidden, but that's where they are. They are due November 4th, which I believe is a week from today. So you have some time to do that. Let's see. I think that's everything I had to say about the exams. Again, I was impressed. I could tell those of you that studied and those of you that did not study, there was a very clear demarcation. So hopefully you see your score. If you like it, great. If you don't, you know some study habits to pick up for next time. This is a little bit of a longer unit, so you shouldn't feel as rushed. All right. So, So before fall break, we talked about photosynthesis. And I know that's probably not what you want to talk about after taking an exam on it. But I love photosynthesis. And I really wish we had more time to talk about photosynthesis. So I wanted to give you a few slides on how photosynthesis can vary. I am not going to test you on this, all right? Because we're just not going to go into enough detail that I don't think you'll have a full understanding of it enough for me to expect you to know this for an exam. But I just want to let you know that variations exist. They are out there. All right, if we're thinking about variations and why they exist, it has to deal with this process called photorespiration. All right, so if you remember the Kelvin cycle, right? The Kelvin cycle takes in carbon dioxide and we make sugars. Right? When carbon dioxide comes in, it combines with RUBP to make 3-phosphoglycerate. And the enzyme that facilitates this is called rubisco. Right? Rubisco is thought to be the most abundant enzyme on Earth. So normally, rubisco is going to facilitate carbon dioxide combining with RUBP, and we're going to make 2 3-PG molecules. That's what normally happens. That's the type of photosynthesis we talked about before break. Right? Now when this happens, the first molecule made, this 3-PG, has 3 carbons. So plants that use this type of photosynthesis, we call them C3 plants, because that first intermediate having 3 carbons. Right? Most plants are C3 plants. Most plants are going to function exactly how we talked about in class before break. Right? I put an example of an oak tree up here. Right? If I had randomly pointed to a plant outside, 90% chance that it's going to be a C3 plant. Right? 90% of all plants on Earth are C3 plants. Right? So this is what normally happens. Now sometimes rubisco can actually add in an oxygen. Right? So normally rubisco adds in carbon dioxide. Right? But sometimes it can use oxygen instead of carbon dioxide. And I know I'm going a little fast here, but I'm not going to test you on this. Right? So rubisco adds oxygen to the RUBP instead of CO2 in certain circumstances. And when this happens, we still get 3-PG, but it only yields 1, so the yield is lower. And it also produces this extra molecule, 2-phosphoglycolate, which this molecule actually releases carbon dioxide and uses up ATP. It is energetically very unfavorable for rubisco to use oxygen. Right? Now normally this is not a problem. Normally rubisco is going to use carbon dioxide. In fact, it wants to use carbon dioxide. It has a higher affinity for carbon dioxide than oxygen. So given the choice, it's going to choose carbon dioxide to use instead of oxygen. But there are certain instances where there is no longer enough carbon dioxide and it has to use oxygen. And that's what leads to this process called photorespiration. All right, this process, photorespiration, typically occurs in plants that live in hot and dry environments. So if we think about our plants and we think about our stomata. Remember, the stomata are the openings on the leaves. That's where carbon dioxide can enter in. That's where oxygen can leave. Right? But because it's an opening on a leaf, that means water can also evaporate out of the leaf through those openings. Right? So normally those stomata are going to be open during the day and closed at night. That's typically what most plants do. Not all plants, but most plants. Now plants that live in really hot and dry environments, right? If those stomata are open, they're losing a lot of water. And if they live, say, in the desert, they don't have a lot of water to begin with. So plants have a dilemma. They have a choice that they have to make, right? They could either keep their stomata open and risk death from dehydration, right? All that water is going to evaporate out of those stomata. or they can close the stomata. But by closing the stomata, they halt photosynthesis. The Kelvin cycle can no longer run because the stomata is closed and carbon dioxide can no longer enter. Right? Now, if the stomata close, carbon dioxide can no longer enter. So we're cutting off the supply of carbon dioxide, which Rubisco likes. The light reactions can still occur, and the light reactions produce oxygen. So now we get a buildup of oxygen inside of the leaves. So at this point, Rubisco will now use that oxygen and go through photorespiration, which again, super inefficient. We don't want that to happen. So plants that are prone to photorespiration, that live in hot and dry environments, have ways to combat photorespiration, to make sure it doesn't happen. And this is a very short and sweet version of how they combat this, right? I'm not going to go into any sort of detail here. They could be a C4 plant, right? A C4 plant, again, named because of the number of carbon molecules in its first intermediate. They actually spatially separate where processes occur, right? So they don't have everything happen in a mesophyll cell. They have parts of photosynthesis happen in a mesophyll cell, but then they have another type of cell called a bundle sheath cell, where photosynthesis also occurs, right? So carbon dioxide comes into the mesophyll cell, and it's essentially going to be accumulated in that bundle sheath cell. And that bundle sheath cell is where Kelvin cycle occurs, where Rubisco is at. So it ensures we always have a high supply of carbon dioxide in that specific cell. So we're spatially separating where processes happen. You could also be a CAM plant, right? A CAM plant, that stands for crassulacean acid metabolism. It was named after the family of plants that this was first discovered in, the Crassulaceae, right? They use a different method. Instead of spatially separating out, they temporally separate out when things occur, right? So now a CAM plant is going to utilize the day and the night cycle. At night, they're going to open their stomata, right? At nighttime, when it's cooler out, when they're not going to lose as much water to evaporation, they're going to open their stomata, take in carbon dioxide, store that carbon dioxide, concentrate that carbon dioxide, so in the daytime, it can close the stomata, run the Kelvin cycle, and have this storage of carbon dioxide it can rely on. That way Rubisco does not have to use oxygen. All right? So, big picture. Variations exist. Variations exist because we don't want photorespiration to occur. And there are different types of variations. We can temporally separate out processes or spatially separate out processes. And that's about the extent that I'm going to go into variations, right? Now, I am a little sad that we don't have time to go into this in more depth. So if you want to learn more, come talk to me. I love talking plant physiology and the weird way that plants work. You could watch my lecture from last year, or there's a couple of shorter videos that you could watch if you're curious as well. All right, any questions? All right, we're going to move into our next unit then, and this is part of what you will be tested on. So this next unit revolves around DNA, right? DNA and RNA, how it's replicated, how it's passed on. So we're also going to talk about mitosis and meiosis here, as well as transcription and translation. How are we using DNA to make proteins? So again, there's a lot of processes, and there's a lot of terms as well, right? This particular lecture is a little heavy on terms, so I'm going to do my best to make sure we're all following along, but I would highly recommend reviewing this lecture afterwards, because all the terminology we're going to talk about today is going to come back time and time again in the next few lectures. All right, so let's talk about our genetic material. First of all, what is genetic material? What makes a molecule genetic material? Well, there's 4 different criteria, right? The first of which is that it has to contain information, right? It has to contain some sort of information that can be used, right? So in our DNA contains information about which proteins need to be made, or how genes are expressed. It also has to be replicated. right? So we have to be able to take one molecule of DNA and make an identical molecule of DNA. It has to be replicated. And we'll talk about DNA replication. I was hoping to start on it today. We're not going to get to it today. We'll talk about it on Thursday. Right? It has to be passed along. It has to have some sort of transmission. All right? So the DNA that is in our cell, when that cell goes to divide, it has to pass along that DNA to the newly created cells. Or an organism needs to be able to pass along its DNA to its offspring. Right? It has to be passed along. And then finally, it has to have the ability to contain variation. Right? So it contains information, and maybe it contains information on skin color. Right? But it might contain different information. It might contain variations on skin color. Right? So it has to have the ability to contain some variation. All right, so it contains information. It can be replicated. It can be passed along to a new cell or a new organism. And it can contain variation. That's what makes it genetic material. Right? If it meets those criteria, we can say, yes, this is a type of genetic material. I like this photo that I put up here, just a quick aside. This is actually an image of DNA that was taken back in 2012. Which is pretty cool that we have an image of DNA. It looks very fuzzy, but this is, we're talking a really small molecule here. So hopefully by the end of lecture, you'll be able to look at this image and understand what all these arrows are pointing to. All right, so we have our genetic material. We know what makes genetic material. All living organisms are going to use DNA as its genetic material. All right, so that includes bacteria, archaea, plants, animals, insects, reptiles, fungi, anything that's living, right, is going to use DNA. Now I did put on here an exception, right? Viruses are not considered living, right? They don't have the ability to reproduce and they're not made out of cells. So viruses don't have DNA, but they still use a type of genetic material. They actually use RNA, right? But all living organisms that we consider living, composed of cells, they can reproduce, they can respond to stimuli, right? All of those different definitions, they use DNA. So DNA is going to be our focus. I want to make sure I'm not going too fast. All right, so we have DNA. It's our genetic material, contains information, can be replicated, passed along, and contain variation. All living organisms have DNA. We're going to focus on DNA, but we're also going to talk a little bit about RNA, because RNA plays a role in how we read the DNA in order to make proteins, or in order to express different genes. So in today's lecture, we're really going to go over the structure of RNA and DNA. And if we look at the levels of structure, DNA has many more levels of structure than RNA, right? RNA and DNA, they both have nucleotides. Okay, we've learned about nucleotides. We'll do a quick review on nucleotides in a second. Those nucleotides will polymerize, and they will become a strand, right? Both DNA and RNA have strands. For RNA, this is kind of the end of the road in terms of level of structure. For DNA, we can continue on, because now DNA can have two strands that form a double helix. They will form chromosomes and make up our genome. And we'll talk about all of those terms. But I want us to review nucleotides. We've talked about nucleotides, but it was all the way back in Unit 1. So I want to jog your memory with this review slide here. Right? I want you to answer these questions, and we will talk about them in a couple minutes. I'm actually going to pass out some content checks that I have not gone back to yet.
Nucleic acid. Monomer is composed of, there's glucose, phosphate group, and then because it's got the little string of three phosphates, it's got the little house, and then it's got the one string.
It's a good way to remember it. How it's DNA different from RNA, just like DNA, because it's two strands.
Whoa, you know that?
I think that's
That's at least one of the reasons I guess. DNA can become RNA.
Yes.
No, I got 2 out of 2. I'm scarring my nose. It keeps like opening up. It's been like that for years since like 1 week. Well, I used to have a nose ring. Yes. I don't have a nose ring anymore because I used to have but not like straight through. It was like it was like sideways in my nose. So it's like a
I thought you were like, yeah, I just
no.
Look at me, I'm very professional.
I would really do. I used to want to
Did you ever make the button?
no, but that's
All right, I'm gonna actually take it back because it's Halloween. What's going on? Because I didn't know.
I'm gonna dress up as for concert with my buddies. Yeah, they're adoring.
So, where's the money?
All right, so hopefully this is jogging your memory. All right, you have to dig deep into the recesses of your mind. Back to unit 1. All right, if we're thinking about DNA and RNA, what is the monomer called? Nucleotide. Yes, it is called a nucleotide. Right? We put many nucleotides together, we get a polymer. What do we call a polymer? A nucleic acid. We could also say DNA and RNA. Right? So if we're looking at our nucleotide, it's composed of 3 basic units. Right? What is this first unit with the P and the O's? It's the phosphate group. All right, we have this next unit down below. It's this pentose shape here. It's a sugar, a pentose sugar, right? And then we have our final part of our nucleotide. It's a nitrogenous base, yes. So all nucleotides, regardless if they are from DNA or RNA, have these 3 structural components. We'll talk about how they're different between the 2. All right, so if we're thinking about DNA versus RNA, how is DNA different from RNA? What's one difference?
DNA is like in that double-stranded double helix, while RNA is like single.
Yeah, DNA is double-stranded, RNA is single-stranded. What's another difference?
DNA uses deoxyribose and RNA uses ribose.
Yes, they use a different type of sugar.
RNA uses uracil instead of thymine.
Yes, DNA uses thymine instead of uracil. What else? What about, yes, where they are. Yes, so DNA is stored in the nucleus where RNA can leave the nucleus. Yeah, those are kind of those key differences, right? DNA is double-stranded. It's a double helix. It stays in the nucleus. It uses a different sugar and can use a different nitrogenous base as well. All right, any questions before we move on? All right. let's take a little bit deeper of a dive into how nucleotides differ between RNA and DNA. So, we mentioned these already, right? DNA has a different type of sugar and uses a different nitrogenous base. So DNA uses deoxyribose as its sugar. And we'll talk more about bases in the next slide or two here, right? But it can use thymine as its base, whereas RNA cannot. Right, RNA, on the other hand, again, different type of sugar, uses a ribose instead of a deoxyribose, and has the ability to use uracil as a nitrogenous base instead of thymine. Right, so those are the key structural differences between the nucleotides in RNA versus DNA. Now, we use a different sugar, and I wanted to make sure I told you why we use this different sugar. If we look at the difference in the sugar here, right? Ribose contains this hydroxyl group. It contains an OH group, right? Whereas deoxyribose does not. And this is the difference. This is why the difference matters, right? This hydroxyl group is very reactive. It's a little less unstable, right? For RNA, it doesn't matter as much. But for DNA, that stays in the nucleus. Kind of think of it as like your passcode, something you don't want changed or messed with. You want it to be very stable, right? It doesn't have this hydroxyl group, which makes it more stable, right? Which is why we use deoxyribose in DNA. Now, RNA has the ability to be made from DNA as a template. We'll talk about that process. It can leave the nucleus, right? And it encodes information that will make proteins. It's shorter lived, right? It doesn't need to be as stable because it's a shorter lived molecule. And it needs to be able to be reactive, right? It needs to be useful. We need to use that RNA to make proteins. So this hydroxyl group is actually better for the RNAs because it makes them more useful. All right, so RNA is useful, DNA is stable. And that's why we have our difference in sugars. All right. So we have our difference in sugars. We also talked about our difference in our nitrogenous bases. So we're going to elaborate on our nitrogenous bases. Back in Unit 1, I told you you did not have to memorize the nitrogenous bases. This unit, you do have to memorize the nitrogenous bases. And you have to remember how we group our nitrogenous bases. All right, so our nitrogenous bases can be placed into two different categories: the pyrimidines and the purines. Now they're grouped based on their shape. The pyrimidines only have a single ring structure. So if we look at the image, the top row are our pyrimidines, right? They only have a single ring. That would be cytosine. That would be uracil in RNA and thymine in DNA. Right? They're all grouped together because of their similarities in their shapes. They are the pyrimidines. On the other end, we have their purines. And our purines are our double ring structure. So that would be guanine and adenine, right? They have two rings associated with their structure. All right, so purines versus pyrimidines. Make sure you know which nitrogenous base falls into which category. Before we move on, I wanted to talk about why we even have this weird uracil thymine thing, right? DNA uses thymine, RNA uses uracil. Why is this? All right, I'm going to walk us through this in the most simplistic way that I can manage. All right? So, first of all, thymine is energetically expensive because it contains this extra group, this methyl group, right? That's more building blocks that we need, more atoms that we need to include into our molecules, more bonds that have to be made. This is expensive to do, right? This takes energy to do. So it's more expensive to make. All right, so that's going to play a key role. DNA is a longer-lived molecule. It stays in the nucleus, right? It makes sense that we would want to invest a little bit more energy into a molecule that's longer-lived, right? So DNA can use thymine. RNA is short-lived, right? It's made from DNA, it leaves the nucleus, it encodes the protein, then it's done, right? Because it's short-lived, we don't want to invest as much energy into it, so we can use uracil instead. All right, so that's one of the reasons. The next reason has to actually do with a totally different nitrogenous base, all right? So we have our cytosine, our other pyrimidine here. Cytosine actually has the ability to become uracil, right? It has like this half-life, so over time it's going to degrade into uracil, right? Now, typically this is not a problem, right? Our cells know that this happens, and we are able to fix it when this occurs, right? So this is typically fixed. Now, this is where the cleverness of uracil versus thymine comes into play, right? DNA uses thymine. It also has cytosine. So if cytosine were to spontaneously become uracil, that right there is a clear indicator uracil should not be in DNA. We need to fix this, right? So that's a good key to our cell's mechanisms to say that this is an error, this needs to be reverted back to cytosine, right? And DNA is a longer-lived molecule, so this can occur in DNA. Now RNA is a shorter-lived molecule. In fact, RNA is so short-lived that typically we're not going to see cytosine turn into uracil, right? It just doesn't happen in that short of a time span, right? Cytosine typically is not going to turn into uracil in the lifespan of an RNA molecule. All right, well, that's fine. That means we don't have to fix anything, but it also means we can use uracil in RNA because it's cheaper. There's no risk in using the uracil, right? If DNA used uracil, and cytosine spontaneously became uracil, right? And a fixing mechanism came along and saw two uracils, right? It would not know which one was supposed to be cytosine, right? And which one was supposed to be uracil in the first place, which is why DNA uses the thymine, right? It's that extra protective mechanism. All right. Does that make sense? Are there any questions there? All right, it's very fascinating and it's very clever. All right, so before we move on to talking about strands of nucleotides then, I want us to do yet another review of terms that we have already learned, right? And I'll give us another couple of minutes.
Isn't a sugar
Good job.
11th grade bio, or 9th grade bio. Romantic.
Wine and dine.
Marriage. I don't fucking know.
This is excellent. Is that what that sounds like? Or is this like Covalent vinyl. Let's go with and
No, not that.
oh yeah. It uses a lot of energy. Yeah. Yeah. Let's guess that. And then
So I guess it's probably covalent.
oh.
What?
What's the name of this specific
Probably. I think you're just.
No.
All right, these terms might have been hidden a little bit further back in the recesses of your mind. All right, so let's review them. We are talking about the backbone of a nucleic acid. This can be DNA or RNA. What is the name of that backbone? The sugar phosphate backbone, yes. So we talked about this backbone. It's a sugar phosphate backbone because it contains the sugars and the phosphates of our nucleotides. All right, when we join two nucleotides together, what type of chemical reaction takes place? It's either a dehydration or a hydrolysis reaction. It is a dehydration reaction, yes. Dehydration reaction takes place, right? So a molecule of water is leaving the reaction, and what's left behind is a bond in its place, right? And that bond that's left behind, it's a covalent bond, and it has a very specific name when we talk about a covalent bond between two nucleotides. What is that name? It's actually on the screen.
I was about to say that, I didn't want to say it.
Maybe no one wants to try to pronounce it. It's a phosphodiester bond, yes. Right, on the screen it's called a phosphodiester linkage. It's the same thing, phosphodiester bond, right? The covalent bond between two nucleotides. All right, are there any questions about the components of how a nucleotide becomes a nucleic acid, this polymerization process? All right, we're going to elaborate on what we already know then. All right, so our strands of nucleotides. One thing we did not talk about, or not talk about in that much detail in Unit 1, is that these strands have a directionality. Right? So when we look at a strand of DNA, of RNA, right, and say we're scientists, we're using this specific sequence, we want to communicate this specific sequence to our colleagues so they can use the same sequence, we need to write it down. We're going to write it down so that it has directionality, right? Now typically, we're going to write it from a 5' to a 3' end. Now we'll start to see some exceptions to this when we move on in our terminology here. But what that means is that we are paying attention to the specific carbons in the sugar molecule, right? So we have our deoxyribose in DNA here. We have numbered each of the carbons, starting on the right-hand side, right? So this rightmost carbon is the first prime, or 1 prime. Going down, we have 2 prime, 3 prime, completely across from the 1, we have 4 prime, and we have 5 prime. Now, when we look at a strand of DNA, we're really going to focus on the 5 prime and the 3 prime, because those are the carbons that are linking the nucleotides together. Right? So if we look at this strand, we have our 3 prime carbon, we have our 5 prime carbon, and in between, they are linked together via this phosphate group. Right? There's a phosphate group in between them. Right? So when we write down the strand of DNA that we are working with, we're going to say that one end is the 5 prime end, and one end is the 3' end, right? So we'd have to look at our molecule, right? And if we look, the 5' end is over here. This one's nicely labeled for us. And the 3' end is at the bottom. Now a trick that I have, and I actually put this on the next few slides, and I'm going to tell you this trick now because I think it's helpful. If we look at the sugar is going to have an oxygen. That oxygen on the sugar is always going to point towards the 5' end. So in here, all of our sugars are on the top, which means the top of this molecule is going to be our 5' end. So if it wasn't numbered, that's how we would tell. All right? I want you to practice this. All right? I want you to practice numbering your strand. Then I also want you to think back. There's some good mnemonics out on Google, or maybe you have one of yourself that you've come up with. How are you going to remember which are the purines and which are the pyrimidions? I'll give you a few minutes.
Would it be directional?
Oh, yeah.
Like what we go back.
I can't remember the pure is gold, I could remember the check.
Oh, because it's 5. No. I wasn't listening. I don't like those. I just don't. It doesn't.
It's a bit confusing. It's close enough.
This one is like, so like, it's
Like it makes sense. Like you're 5. So it does start here. You're 5. You're 3. You're 5. And also you're 5. Like this is the 5 question here. Here. Whereas like you say, this is the 5. And something like this. Left. And this 5. This 5. Here. Yes, that's the 5. So like you're always bottom right. Which the bottom right. You're always 3. This is always 5. So it's
oh, but it matters what you
okay. 5. Left.
But the order is in.
Like the 5 is included in here, which is on the bottom way.
That's how it's for.
Right.
The order tells you.
Like you're always with 5. So this is for the first 5 that's used. 5. 5. It would be for the 5. That's why 5. Like it points the way it's going. Because the 5 always because the 5 always the 5 is always the top left. It's always with 5. So the way you move the direction is always the way. Because it's always going to the way that the
All right, does anybody have any way to remember the purines versus the pyrimidions? Yeah, that's a great one. In fact, if you go back to the slide where I talked about purines and pyrimidions, in the notes, I've actually linked that one. But pure as gold. So pure for the purines, as adenine, gold, guanine. The other one being cut, C-U-T, the pi for pyrimidions. That's a way you can remember it. Does anybody have another mnemonic? Or a way to remember this? I'll tell you mine. Mine's not as like fancy. Mine's just the shorter word belongs to the larger molecule, and the larger word belongs to the shorter molecule. That's how I remember it. To each their own. All right. No one else wants to share their mnemonic. We'll move on. If I were to write down the sequence of this DNA strand, how would I start this? What is the first thing I would write? I would write 5. Yeah, I have to indicate directionality, and I'm going to start by writing the 5. I'm going to start at the 5' end. All right, I'm going to start with the 5. Now I'm going to talk about my nucleotides. So which is the first one that I'd write down? T, because it's at my 5' end. Right, and then you'd write G, A, C as they appear. And then how am I going to end this? By saying 3'. Yes, I want to indicate the directionality. Right, this is very important. And we are going to see this come up many, many times. All right, so if I'm writing down just a single strand, I'm always going to write it 5 to 3. Now if we're writing down multiple strands, or we're talking about the opposite strands, I might write it 3 to 5. But for just a single strand, you're going to write it 5 to 3. That's a good rule of thumb. Any questions? All right, well, we talked about single strands. Now let's talk about 2 strands put together. All right, so at this point, we're only talking about DNA, because only DNA has a double helix, has 2 strands. All right, so DNA has a double helix, and it's 2 molecules that are bonded together. So we have one strand, which is one molecule. We have another strand, which is another molecule. And they are bonded together using hydrogen bonds. And we'll see that in the next few slides here. Typically, a lot of these visuals that you will see, how the artist represents these bonds are going to be dashed lines, which you can see on this image here. That indicates hydrogen bonds, most of the time, not all the time. Some artists do it in different ways. All right, so we have 2 strands. 2 molecules bonded together via hydrogen bonds. Right? We're getting our DNA. This is where our base pairs come into play, complementary base pairing. So we have our nitrogenous bases, our C's, our T's, our A's, our G's. Right? They're always going to pair in a very specific way. A is always going to pair with T. Right? And G is always going to pair with C. That's always going to be the case. Right now, a way to remember this, I had a student in my last class teach me this one, where you can say an apple falls from the tree, so A and T, or the car gets parked in the garage for C and G. I like to remember it as the curved letters, G and C, go together. But if we're looking at these specific pairings, you should notice that it's always a purine connected to a pyrimidine. Right? So our purine, our adenine, is always connected to the pyrimidine of thymine. Same for guanine and cytosine. Now, why are they always connected in this manner? Well, first of all, a purine is always going to be connected to a pyrimidine to maintain a constant shape. Right? So we have a large molecule and we have a small molecule. Right? If we constantly have a large and a small, we're going to have the same shape, this constant structure, the entire way down our molecule. Now, if we have a pyrimidine, let's see, pyrimidine would be the small one, with a pyrimidine, right? It would narrow a bit. It would change our structure. Or if we have a purine and a purine, it would widen out because those molecules are both big molecules. So we'd have something that's very like wobbly, has no uniform structure to it. right? So we're always going to pair a pyrimidine with a purine to always have a very unified structure. Right? But that's not the only reason. It's actually already on the screen. All right, the other reason is because of the hydrogen bonds, the way that the hydrogen bonds work, right? Adenine and thymine can each make 2 hydrogen bonds, so they're complementary to each other. Whereas guanine and cytosine can each make 3 hydrogen bonds, so they are complementary to each other, right? Just based on the structure of the molecules themselves. Right, so that's why we have this complementary base pairing between purines and pyrimidines, and why C always goes with G and A always goes with T. All right, so if we just pair them up into 2 strands, right, so they look parallel to each other, we can also look at directionality. All right, so again, this is where that clue comes into play. Oxygen points to the 5' end. So on one side of our strand, our left side here, oxygen is at the top of the sugar, right? So it's pointing towards the top. We know that's the 5' end. Opposite, it looks like this molecule has been flipped around, right? Now oxygen is pointing down. So we know at the bottom is the 5' end. Right, so we notice that our 2 strands, right, they have directionality and they run opposite to one another. We would call these anti-parallel strands, right? They're parallel, but they're anti-parallel, meaning they're running in 2 different directions. Right, again, directionality playing a huge role here. All right. Were we ready to move on? I felt like I switched that pretty fast. Give it a few more seconds here. So this in and of itself, right, pairing our 2 strands together, doesn't necessarily make it a double helix. Right? And it's drawn not as a double helix on this screen. There are other factors in play that actually give it that twisting structure. And I'm not going to go into too much detail because it's kind of complex, right? But that helix structure is formed due to a variety of reasons. Due to how we can most efficiently stack our nucleotides, due to its interactions with water, there's parts of this molecule that doesn't want to interact with water, and due to the geometry of the sugar itself, all leads it to have that twisting structure. Right? Now, I'm not going to get into much detail here because it is more complicated and more than what I would expect you to know in a Bio 110 class. But if you are curious, I have linked a video at the bottom that does a really good job of explaining why it twists. Right? So once it twists, once it becomes this helix structure, right, we can specify this helix structure even further by saying it's a right-handed helix. Right? So that indicates the direction in which it twists. Now, I have a really hard time visualizing this in my head. I have to go through and use this staircase analogy. So if I have a staircase, right, a spiral staircase, has to be spiral. So there's railing only on one side, and it's going to be on the outside. If I go up the staircase, and I'm going to use the railing, so I'm going to say on this staircase here, I'm going to use my right hand on the railing. That would be a right-handed helix, right? Now, if I use my left hand, right, turning the opposite direction, that would be a left-handed helix, right? So I have a really hard time visualizing this. There's a method that, yes, you can do with your thumbs that helps explain it as well. But I want you to know it's a right-handed helix. It has some handedness to it. All right? Not only is it right-handed, but there are features within the helix itself that'll play an important role in conceptual learning upper-level biology classes. But for now, I want you to know that the helix has these grooves. So the way that it twists gives it 2 types of grooves. A major groove which is the larger groove, and a minor groove, which is the smaller groove. Now the grooves have the same basic function, right? These grooves function as a place for proteins to bind, right? So when you learn about other processes involving gene expression, right, proteins can bind to the DNA and it's going to bind in these grooves. The major difference here is just the size of the grooves. The bigger the groove, the more places to bind. The smaller the groove, the fewer places to bind. All right, so they have grooves, one smaller than the other, and that's where proteins can bind. And that's all the depth we're going to go into for the grooves. All right, let's do some practice. I want you to label the directionality on my DNA helix below. Helix that has been flattened, not into a helix, as it's drawn. And then write the complementary sequence to what is on the screen.
Is this just this is is that
Yeah.
that's great. Okay, great. Okay. It's double, so you need two.
Exactly.
I feel like I might be just gonna have that. Yeah, you have it. Exactly, that's
My rabbit is on Edsa. I bet he's blind. He ran into stuff he wasn't blind. No, he's only 5 and a half. I think like 8 to 15 years. But they're also very easy to kill.
Oh, that's how it's set up. I can double check and lab
No, I mean probably you could probably do that. If you put them in the bags with it too much, it's not. But you can yes.
Yes.
Also, if you put them in a bag, some people just die. With the culverts one, they get too scared. If they get the rabbit, which is adorable. They die tonight.
Sneak past.
No.
I like that you're turning around and having this great conversation. I have it.
No. So I had a rabbit and we brought him to the zoo then because we
All right, I have given you a portion of a double helix here, and I asked you to label the directionality.
and then they lost the mom.
If I start at the top left-hand corner here, what end would I label this as? 3 or 5? 5 prime, yes. Which means the opposite end has to be the 3 prime. All right, so if we go to the other strand, this top corner, this top right corner, what would I label this as? 3 prime. Yeah, and then the bottom has to be a 5 prime. And hopefully you use that oxygen trick, right? So oxygen is upwards, so we know the top of this strand is the 5 prime. And on the opposite strand, the one on the right, the oxygen is at the bottom. It's pointing towards the bottom. We know the bottom is the 5 prime. All right. I want to write down the complement to this strand. All right. I want to write down the strand that is pairing with the strand I have shown. What am I going to start with? I'm going to start with 3, because remember it's anti-parallel. This is where the exception comes into place. If it's just one strand, I always write 5 to 3. But now I'm looking at a complementary strand that is anti-parallel. I'm going to start by writing a 3. Right? What's going to come after the 3? T, and then T, and then A, and then C and C, and then G and G. Yes, and then I'm going to end it with my 5, right? Because I want to indicate that directionality. When we talk about DNA replication, that's going to be really important. That's why we indicate directionality. All right, my last question up there, how many molecules make up a double helix? 2. Yes, 2 strands. Each strand is a molecule. They make up a double helix. They are bonded together with those hydrogen bonds. All right, any questions before we move forward? All right, so we talked about nucleotides, we talked about strands, we talked about double strands, and the double helix. So next, we're going to talk about our next unit of structure, right, which is a chromosome. And I'll flip to that here, right? But we have to kind of preface this next level of structure by saying that DNA is a huge molecule, right? DNA is long. In fact, if you took one molecule of DNA, so one double strand, laid it out, stretched it all out, it would typically reach around like 1 meter, or roughly 3 feet, right? It's pretty long. So we have to take that strand of DNA and condense it to fit into a cell. And a typical cell is like 10 to 100 microns. All right, so let's think about what a micron is. We have a centimeter, right? 1/10 of a centimeter is 1 millimeter. And we can break that little sliver, that 1/10 of a millimeter, into 1,000 sections. Right? That would be the microns. Right? So 1,000 microns make up 1 millimeter. So if we're thinking about a cell that's about 10 to 100 microns across, that is like the tiniest little sliver of a millimeter. That's really tiny. We have to fit 3 feet of a molecule into that. I can't even put it in my fingers because it's so tiny. To that really, really tiny cell, right? Not only that, DNA has to fit inside of the nucleus of that cell. So the nucleus is even smaller. Nucleus tends to be around 4 microns, right? So in order to fit this huge molecule into such a tiny space, we have to condense it, and we have to condense it down a lot, right? And this is where chromosomes come into play. And I like this fun fact here, that if you took all of the DNA molecules in our body, so every cell, every DNA molecule within every cell in our body, stretched it out, you could go to the moon and back 6,000 times, right? We have a lot of DNA, right? There's some good mileage there. All right, so we have to condense it down so it fits inside the nucleus of a cell, right? So how we do this is that we supercoil this DNA, or we highly condense it down into this form called a chromosome, right? So a chromosome is typically, when you look at a chromosome, you're going to see that classic X image, right? It's not always going to look like an X, but that's typically the most charismatic example of a chromosome that is often depicted in biology textbooks, right? That is all that 1 meter of DNA in that tiny little space, right? The chromosome is not just DNA though, right? It's DNA and it's also proteins, right? So it's DNA, it's wrapped around proteins, it's all condensed into a chromosome. So that chromosome, we would say, is composed of chromatin, and chromatin being the term for a DNA protein complex. So DNA wrapped around proteins. Right, so chromosome, discrete unit of genetic material. We actually have multiple chromosomes, right? All of our DNA does not fit into one chromosome. We actually have 23 sets of chromosomes. The chromosome itself is not just DNA, it's DNA and proteins together in a complex we call chromatin. All right, so now let's talk about how we get it this small. How does it condense into a chromosome? I still see some pens writing, so I'm going to pause a little bit here. All right, we're taking this long molecule, we're condensing it down, and we're doing that in 3 different ways. There are 3 levels of compaction that DNA has to go through in order to be small enough to fit inside of a cell, inside of the nucleus of a cell. The first level is called DNA wrapping. Right? This is where DNA is wrapped around these proteins called histones, and together, DNA and the histones form a nucleosome. So if I'm looking at this image, right, DNA is my red ribbon that's depicted here. It's wrapped around these pink, they kind of look like gravel pieces all clumped together, right? These pink histones, right? And when DNA is wrapped around the histones, this complex all together we call a nucleosome. All right, so by wrapping DNA around those histones, that condenses down our DNA by 7-fold. Right, so our DNA molecule is folding and it's getting smaller. But it's still not small enough. All right, there is still another level of compaction that it goes through. All right, so first level, we have created these nucleosomes. We have wrapped them around proteins, created a nucleosome. The second level of compaction, we call a 30 nanometer fiber. Right? Now it's called a 30 nanometer fiber because that is the width of this structure. Right? But that's where we take nucleosomes and we start to stack them in this very efficient zigzag pattern. So instead of all spread out in a line, now we're stacking them up. Right? So it's something like beads on a string again. Right? I could put all my nucleosomes in a row. Let's see, how many do I have here? 1, 2, 3, 4, 5, 6, 7, 8. Or I could go 1, 2, 3, 4, 5, 6, 7, 8. Zigzag them, and it's going to be compacted down even more. Right? So the zigzag feature condenses that DNA another 7-fold. Right? But that's still not enough. Right? DNA still goes through another level of compaction to reach its final form in a chromosome. All right, so we have our nucleosomes, DNA wrapped around the histones. Our nucleosomes have been placed in the zigzag pattern, this very space-efficient pattern. Now we're going to take this zigzag pattern and loop it. Right? So this is our last level, and we call these radial loop domains. That's the interactions between the 30 millimeter fiber and the proteins to form loops. All right, so I have my zigzag, and I could put it in a line, or I could take my zigzag and loop it to make it smaller. But I'm not just making one loop, right? I'm making several loops on top of each other. Right, so I'm condensing it even further. And you can see that really well in this image here. We have our chromosome. If we were to slice through the chromosome and kind of look down at it, you would see it's all twisted. Right? These are all these radial loop domains. Again, trying to fit in more DNA in a smaller space. All right. So we've compacted our DNA, but not all DNA is going to be compacted to the same level. Right? DNA can be more compacted, it can be less compacted. It's always going to be compacted, but it's going to vary in how compacted. Right? So most of the time, DNA is going to be compact, but not super compact. We're going to call this euchromatin. So if I were to draw a cell, and I'm going to draw a cell that's getting ready to divide, so I'm going to draw a classic X shape. If I draw it as euchromatin, I might draw something long, squiggly, it's not really condensed down. It's an X, it's there, but it's less defined. So it's compacted, but it's not super compact. that would be our euchromatin. Right? This is where the way that DNA is typically housed. Right? If you look at a cell that's not going through cell division, it's typically going to be euchromatin. But when it does go through cell division, when it gets ready to divide, so we're ready to go through mitosis or meiosis, we'll talk about next week, it's going to get even more compacted. And this is where we get our classic X shape. And this would be our heterochromatin. Right, so DNA always going to be compacted, but the degree to which the compaction occurs can vary. All right. Let's do a review. I have left some blanks in this image. I want you to fill in these blanks with the right terminology that we talked about for DNA compaction. It does have a name. It's based on the width. Yeah. Nana, how's it going? Getting it down. Slowly but surely. Yeah. There's a lot of knowledge on this. Yeah.
Especially on how to use like high school, like when I took like art, but it was like there's a lot of stuff that you never like relatively touch.
Yeah. A little bit more in depth. But not nearly as
I disagree. I think that's
Oh, I'm sure.
I feel like I get back from break and my teachers really kick my Chromosome. I have like so many. I was so on campus or sports. And I like to check my classes. When you started, no, I'm in the third grade. Teachers are getting way too
All right, we've taken this strand of DNA and we've compacted it sevenfold into this first level of compaction. What could I label this as? What was that? I could label it as nucleosome. Could I label it as anything else? DNA wrapping. Yeah, I could say it's DNA wrapping. I could say we've made a nucleosomes. This image actually labeled it nucleosomes. Right? All right, so we have our nucleosomes. Now we're going to put it in a zigzag pattern. Right? You can see it's highly compacted in the zigzag pattern here. What would I call this? As a number. 30 nanometer fiber, right? It's based on the width of this level of compaction. All right. I got one more level, right? We're now looping everything together. What could I label this as? I could say we're making some radial loop domains. Yes. This one actually labels it chromatoloops, which is the same thing, right? We're just looping our DNA, right? All right, any questions about compaction? All right, we're going to keep moving on then. So we're still going to talk about chromosomes, right? But now instead of talking about how they're compacted, we're going to talk about some terminology associated with these chromosomes. And this is terminology we're going to see reappear throughout this unit, right? So like I said, DNA is not all in one chromosome. It's spread out across multiple chromosomes. So if we're looking at just one chromosome, we're looking at just a small snapshot or just a little bit of all of the DNA in our bodies, right? Because our DNA is spread out over many. If we're looking at just one, we're looking at just a sliver of the DNA present, right? Now, every single cell contains a full set of DNA, right? So DNA is spread out over many chromosomes. Every cell has all of those chromosomes. Now, if we look at one chromosome, or I'm going to say one pair of chromosomes, there's some terminology associated with them. All right, so I have two different pairs of chromosomes here. One is blue, one is purple, right? You could say one of these pairs, we'll say the blue one, in fact the blue one here says it's the paternal copy, so the blue one came from your dad, purple one came from your mom, right? Now this pair, we would say is a homologous pair, meaning it contains the same type of information. It's not exactly the same, it's not a duplicate copy, but it contains the same type of information. So say this first pair here contained information on hair color, right? Both of these chromosomes in this pair will contain information on hair color. It won't be exactly the same, right? Maybe your mom passed on the gene for brown hair color and your dad passed on the gene for blonde hair color. There's variations, but we're still talking about the same type of information. We're still talking hair color, right? So they're homologous, and that they contain the same information, just different variations of that information, right? Variations that have been passed down from your dad, variations that have been passed down from your mom, right? Same with if we look at yet another homologous pair, right? Maybe this pair contains different genetic information altogether. We're no longer talking hair color. Maybe this one is containing information on your height, right? Again, both contain information on your height, but one comes from mom, one comes from dad. Maybe your dad passed on some tall genes and your mom passed on some short genes, right? There's variations there, but same type of information, right? So these are homologous pairs. Same type of information, different variation. All right, so we're going to elaborate on these homologous pairs. So each chromosome in a pair has some terminology. Now, chromosomes typically look like this. They're kind of like in a straight line. They're not going to be in that X shape unless we're about to duplicate. Most of the time, your chromosomes are going to, if you were to draw it out, look like this straight line, right? So in this homologous pair, we would say the homologous pair on the top or the homologous pair on the bottom, right, contains two chromosomes, right? Again, one from mom, one from dad, same type of genetic information. We could call these chromosomes chromatids, right? It would be the same thing in this case. They're each a chromatid, right? Now, when they duplicate, so when the cell is getting ready to divide, it needs to duplicate its information so it can pass along the information to the next cell, right? It's going to replicate. And now we see that classic X shape, right? That DNA has been replicated. So again, these chromosomes, as a pair, are homologous to each other. But because they've replicated, right, now they contain two copies of all of that genetic information within one chromosome. So they're a homologous pair, right? Each chromosome we could still call a chromosome, right? So this yellow one as an X is a chromosome. Before replication, that yellow one as a singular line is a chromosome. The chromosome that's been replicated has two sides to it. Each side is identical to each other. So we could call each side a chromatid, and because they're identical, we call them sister chromatids, right? They contain the exact same genetic information. It's just been duplicated. Right? So if this one came from mom, now you have 2 copies of mom's genes. Same for dad. Now you have 2 copies of dad's genes, but still in 1 chromosome. All right. Now these 2 sister chromatids that are identical to each other, they're still attached to each other, which gives it this X shape, and they're attached at this location that we call the centromere. That's where they are joined together, is at the centromere. Now I'm introducing this terminology now so that you're aware of it, and we'll mention it a few times, but especially when we get to mitosis and meiosis, this is where it's going to become extremely important to know what is a homologous pair, what is a sister chromatid, what can I call a chromosome, right? And I believe this information comes up on your content check as well. And I'm going to have you practice it right now. So I want you to label this image with the correct terms, and then we'll talk about it.
No.
Oh, no,
3 sisters. Yeah, the one right here. Homologous. Homologous. That makes more sense. Homologous. Duo is I could have draw. That's fine. But over 3 sisters and in school pre-med number. It was like I can only draw. Okay.
Yeah.
They mark in chromosomes. We both start off with a tattoo problem.
But we only do that.
Right? So we can call it
Number 3. I'm not understanding what my dad keeps telling me.
Yeah, that's you look that's like literally that.
Okay. Also, like, what if I on them? I'd rather think of them to progressively worse. Hey, I'm already certified as an EMT. I'm already. If you have a heart attack, I legally have to. That's not how that works. I also don't have my car here. At best, I'm throwing the basket up to my left. And I'm like,
here. Okay. I'm good. So I'm looking at this image, or I give you this image on an exam. We'll label it. What label would you put to number 1? I hear a lot of people talking, so there must be a lot of answers out there. Homologous pairs, yes. Oh, I'm giving you all the answers at once.
oh.
All right, so they are homologous to one another. Right? One came from mom, one came from dad. They contain the same type of genetic information, just different variations on it. All right. Number 2, hopefully you came up with centromere. All right. Centromeres are going to play a role when we talk about how the mitotic spindle connects to the centromeres and pulls them apart in mitosis and meiosis. Right? And then lastly, we have number 3, which I think is the hardest one. Right? Each side of an X is a chromatid, right? But if they're on the same chromosome and they're identical, we call them sister chromatids. All right, any questions? All right. My last slide then is that beyond chromosomes, beyond a singular chromosome, remember how I said our DNA was spread out over multiple chromosomes. This is where we get the term genome. All right, so genome is the complete set of our genetic information. As humans, we have 46 chromosomes in 23 pairs, right? Because we have 23 pairs, homologous pairs of chromosomes, but we have 46 individual chromosomes, right? So 2 chromosomes in each pair. All right, now other organisms have different sets. Other organisms have different types of sex chromosomes. They might be a Z chromosome, or, well, I even forget. There's some wild variations on our chromosomes within the animal kingdom and plant kingdom, right? But humans, we have 23. All right. This is as far as I thought we would get today, so I put the homework slide right here, and I had hoped we'd get further. We're not going to get further. This is where my last class stopped. But I want to say content check number 5 is live. You should be able to answer most of the questions. I think there's like one question you'll have to wait until after class on Thursday to answer. That is due Thursday. There's going to be a lot of terminology in this next class. So if you're worried about content, watch that video in preparation for all the terminology. Exam corrections due November 4th. Scientific literacy due November 4th. This extra assignment due November 5th on the history of DNA. So they are all up on Moodle, all ready to go. That extra assignment is very good.
Okay.
Yes.
Rita, if I give you my test corrections on Thursday, is there any way you could get them in by Friday morning? Because I need my advisor on Friday.
Yes. Can you like write a note on your test corrections?
Yeah.
So it reminds me.
Yeah.
I can grade it early.
Is
I
your
just,
advisor gonna look at you and judge you?
Yes.
Oh my god.
He's a judgy guy, so I gotta be ready.
who's your advisor?
Oh well, I don't have my real advisor yet.
Okay,
So I
you're
have
on
one.
yes?
Yeah.
Yeah.
Mhm.
Got it.
Wow.
He's a little judgy guy, but it's okay. We get along.
That's good.
All right. Thank you.